Inside every single living thing on this planet, from a tiny little bacterium to a massive blue whale, there's a secret code. It's a microscopic blueprint, really, that holds all the instructions needed to build and run that organism. Today, we're going to unpack that code.
So, what is this secret code? What is this incredible molecule that dictates everything from your hair color to your height to how your body even fights off a cold? Well, the answer, of course, is DNA. So let's dive right in.
Probably the best way to think about DNA is to picture a huge, incredibly detailed instruction manual. And I don't mean just one book. I mean a whole library's worth of information. And it's all packed away, super organized, inside the tiny nucleus of pretty much every single cell in your body.
Now, how do you cram all of that information into a space that's microscopically small? Well, the DNA is tightly coiled up and packed into these structures we call chromosomes. So if the DNA in one cell is the entire library, then each chromosome is like one volume on the shelf. For humans, we typically have 46 of these volumes. We get 23 from each parent, and together they tell the complete, unique story of you.
And just how big is this story? Well, the human genome has about 3 billion base pairs. These are the chemical letters of our genetic text. To put that in perspective, if you tried to type all of that out, it would take you pretty much your entire life. The scale of it is just mind-boggling. Okay, so we know it's a massive instruction manual, but what does it actually look like? What's the physical structure? Let's zoom way in and take a look at the most iconic shape in all of biology, the double helix.
Okay, so its fancy name is deoxyribonucleic acid. It's a type of molecule called a polymer, which basically just means it's a long chain made up of repeating units, kind of like a train made of individual cars linked together. In DNA, these cars are called nucleotides, and they form two long chains that hold the fundamental secret of life itself.
The easiest way to picture this whole thing is as a twisted ladder. You've got the two long strands twisted into that classic helix shape. The sides of the ladder, what we call the backbone, are made of repeating sugar and phosphate molecules. But the really important part, the part that holds all the information, that's in the rungs.
And those rungs are built from just 4 chemicals, or bases: adenine, guanine, cytosine, and thymine. We just call them A, G, C, and T. That's it. That's the entire alphabet for the code of life. And what's really clever here is their structure. Some are bigger, some are smaller, and that's absolutely crucial for how they pair up.
And this right here, this is the golden rule of DNA. It's beautiful in its simplicity. The bases don't just pair up randomly. A always pairs with T, and C always pairs with G. No exceptions. It's this simple, perfect lock and key system that allows the code to be read and copied so perfectly every single time. Okay, so we've got a code, and it's written in a 4-letter alphabet. But a book is pretty useless if no one can actually read it, right? So how does the cell take these instructions and use them to build all the stuff that makes you, you?
It's a really elegant 2-step process. First, in what's called transcription, a specific section of DNA, a single gene, is copied onto a temporary message molecule called messenger RNA, or mRNA. Think of it like this. You don't take the priceless original blueprint to the messy construction site. You make a quick photocopy. That's what mRNA is. Then, in translation, a little cellular machine called a ribosome reads that mRNA photocopy and uses it to build a protein.
So how does the ribosome read the message? Well, it doesn't read one letter at a time. It reads the code in 3-letter words called codons. For example, the ribosome might read the codon ACG and know to grab one specific building block, an amino acid. Then it reads the next codon, grabs the next amino acid, and links them together. You string enough of those together in the right order, and poof, you've just built a protein.
So reading the code is one thing, but what happens when a cell needs to divide into 2 new cells? It has to make a perfect copy of the entire 3 billion letter library for that new cell. This process is called replication, and honestly, it's one of the most amazing tricks in all of nature. And this is where that simple base pairing rule becomes so incredibly powerful. First, an enzyme called helicase comes along and literally unzips the 2 strands of the DNA ladder. Then, another enzyme, DNA polymerase, moves along each of the separated strands. It's like a master builder. It sees a T on the old strand, it adds an A to the new one. It sees a G, it adds a C. And the result? 2 perfect, identical copies of the original DNA.
And the accuracy here is it's breathtaking. Just look at this. For the 3 billion or so bases that get copied every time a cell divides, the process initially makes about 30,000 errors. And you might think, whoa, that's a lot. But then the cell's proofreading enzymes kick in. They scan the work, fix the mistakes, and in the end, you're left with maybe 3 typos. 3 mistakes out of 3 billion letters. It is an unbelievably faithful process.
So we've talked about what DNA is and how it works. But what about the so what question? Why does understanding this tiny molecule matter so much in our everyday lives? Well, because it has completely changed our world, unlocking technologies that were basically science fiction just a generation ago.
This is where the blueprint becomes a toolkit. Through genetic engineering, we can program bacteria to become tiny factories that pump out life-saving medicines like insulin. In forensics, DNA profiling allows us to identify individuals with incredible accuracy. With genetic genealogy, we can trace our family trees back hundreds of years. And with bioinformatics, we use supercomputers to analyze all this genetic data to understand and fight complex diseases.
And you know what's wild? How recently we figured all of this out. I mean, DNA was first isolated way back in 1869, but it wasn't until 1953 that Watson and Crick, with absolutely critical data from Rosalind Franklin, finally figured out the double helix structure. And the first full draft of the human genome, that was only published in 2001. It has been an absolute whirlwind of discovery. And that really leaves us with a pretty profound final thought. For the first time in the history of life on Earth, one species, us, can not only read the instruction manual for life, but we're quickly learning how to write and edit it too. The possibilities are staggering, and so are the responsibilities that come with holding that kind of power.